In this presentation, I will talk about association between placental oxygen transport and cerebral cortical growth in fetal brains. Human fetal brain shows very dynamic development process and major neurogenic events, including neurogenesis, proliferation, and migration. We can see very rapid brain cortical surface growth and folding with cortical circuit and axonal development. Because normal cortical growth and folding patterns are important for normal brain function, it's been of great interest to understand developmental mechanism of human cortical growth. Human cortical development is strongly controlled by genetic process. Cortical functional areas and folding patterns are spatially associated with gene expression pattern in ventricular zone and cortical plate. But gene expression associated with brain development can be modified by environmental factors in fetal life. Our previous study and data also showed that genetic is not everything in brain development. When we see 3D brain surface model in monozygotic twins who have the same genotype, their brains look different. They are more similar than unrelated pairs, but their brain structures are still not identical. Even before birth, we can see different brain shape in fetal monozygotic twins. So there must be many other factors influencing fetal brain cortical growth. Among many factors, placental function is a crucial factor for fetal growth. Fetal health is dependent on placental function, especially placental transport of oxygen from mother to fetus. Placental insufficient oxygen transport can result in fetal growth restriction and atypical cardiovascular, metabolic, and neurological development. And this atypical fetal growth can also affect brain development. So our hypothesis is that placental transport of oxygen may be one of the important non-genetic factors that affect early human brain growth. And this study aimed to assess placental oxygen transport function and examine its impact on fetal brain cortical development in vivo using fetal MRI. We performed monochorionic twin brain analysis because twin subjects have identical genotype and gestational age. So we examined the placental transport effects controlling for genetic and maternal factors. To present our study, first I will show in vivo MR imaging of fetal brain structure and placental transport function, and I will talk about our quantification of fetal cortical growth and folding pattern and placental oxygen transport. Then I will show significant relationships between cortical growth and placental function. Our study included 8 monochorionic twin pairs, and their age ranged from 27 to 34 gestational weeks. We scanned fetal brain using single-shot fast spin echo T2 weighted imaging for structural MRI. It is usually necessary to acquire multiple images in axial, coronal, and sagittal views due to interslice motion artifact. To image placental oxygen transport function, we are using bold MRI. We perform bold MRI with maternal hyperoxia, which is transient 100% oxygen inhalation, and it can non-invasively measure blood oxygenation in the placenta. We can see imaging signal change during hyperoxia in placenta region. To quantify fetal cortical growth, we process structural fetal MRI data using our pipeline. From the raw data, we use the UNet deep convolutional neural network to automatically segment only brain regions, and we excluded bad quality imaging data for further reliable data process. And T2-weighted structural fetal MRI shows interslice motion artifacts, so we are using slice to volume matching and registration techniques to remove that interslice motions. After motion correction, we performed cortical plate segmentation. We recently developed automatic fetal cortical segmentation method using 2D fully connected convolutional neural network. From the segmentation volume, we reconstructed the surface model with the same number of triangles and a good smooth mesh property. For whole brain global analysis, we measured cortical volume, cortical surface area, gyrification index, and mean sulcal folding depth in the left and right hemispheres. We performed sulcal regional analysis. Recently, we developed the method for automatic postulation and labeling of cortical sulci in the fetal brain. Using our automatic sulcal labeling method, we measured surface area and sulcal folding depth for each sulcus. We also analyzed the global sulcal folding pattern, which is the patterns of arrangement, number, size, and relationship of sulcal fold. It's biologically meaningful to examine the sulcal folding pattern because it is related to size, organization, and spatial arrangement of cortical functional areas and their white matter connections. And this sulcal pattern variability is related to several human functions and many neurological disorders. For quantitative sulcal pattern analysis, we characterize the sulcal pattern with the graph structure, and this graph has geometric and topological features of sulcal fold. Previously, we developed a method to compare sulcal graphs between different brains using a spectral-based graph matching technique. Then we calculated the normalized sulcal pattern similarity index using combined features of position, depth, and area of sulcal fold. And this index ranges from 0 to 1. We can also use a single feature and measure the sulcal position, depth, and area patterns separately. For quantification of placental oxygen transport, first we manually delineated the placenta regions corresponding to each twin fetus and extracted placental signal change with maternal hyperoxygenation. We employed the gamma function to capture and model the signal change and obtain time to plateau TTP value after fitting the signal to the model. TTP can be a surrogate marker assessing regional oxygen delivery function in the placenta. For healthy subjects, TTP values were short and placental histology was normal, but for pathological cases, TTP values were longer and less uniform and histology was abnormal. We statistically analyzed the relationship between mean placental TTP value and global and sulcal regional cortical growth measures and sulcal pattern similarity between twin pairs. Volume, surface area, gyrification index, and sulcal depth were compared between twins with low TTP and twins with high TTP using a paired t-test. For sulcal pattern analysis, we examined the relationship between sulcal pattern similarity and TTP difference of twin pairs using correlation analysis. The group analysis showed that fetuses with long TTP value showed significantly lower cortical plate volume in the left and right hemispheres. And cortical surface area in the left and right hemispheres were also significantly reduced in the fetuses with high TTP values. Whole brain mean sulcal depth and gyrification index were not statistically different. Our sulcal regional analysis found significantly reduced sulcal depth in the right inferior frontal sulcus and right parietal occipital sulcus in fetuses with high TTP value. In the sulcal pattern analysis, sulcal pattern differences were significantly correlated with TTP differences in twins in the right hemisphere. When TTP difference was larger, sulcal pattern similarity was lower between twin pairs. And we examined each feature separately and found sulcal position and depth patterns were associated with TTP value. In conclusion, we identified placental non-genetic prenatal influences on human cortical development. Alterations in the placental oxygen delivery function caused reduced volumetric and surface growth of the fetal cerebral cortex and altered sulcal folding pattern. And this cortical structural alterations may be associated with the future neurodevelopmental outcomes at the birth. So this study provided meaningful information for understanding developmental mechanisms of the human cerebral cortex. Thank you for your attention.